Good morning, friends. In the last two classes, we understood the discovery of auxin as well as the biosynthesis of auxin. Today, we will understand the signal transduction pathways of auxin. So, for that, we need to understand the proteins involved in this action. So, two category of protein families are involved. One is AUXIAA or AUXIAA. Another is ARF protein. This ARF protein is basically a short-lived nuclear protein, and these can bind to auxin response genes. So this nuclear protein, you heard this word nuclear protein. This means this protein can enter into the nucleus and can bind to the DNA. Okay, so these two proteins exert their regulatory effect by binding with each other and then by binding with the DNA or the auxin response gene or the part of DNA which carry the auxin response genes. Now let us understand what is happening to the pathway when auxin is present as well as when auxin is absent. So to understand that, let's look at this diagram. So the first diagram is about when auxin is absent. So when auxin is absent, this AUXIAA protein and this ARF protein, they will form basically this dimer, and it will enter into the nucleus and it will associated with the auxin response gene. But as the AUX is present with ARF, which is acting as a repressor, it will not help to express the gene. So there will be no gene expression and no response. Okay. And in second case, when auxin is present, what is happening? Now, when auxin is present, AUXIAA protein, which is a repressor, will undergo a degradation process. Let us understand this segment. So, AUXIAA and ARF, they are usually present in the cell in dimer form, in this form. Okay. So, when auxin binds to its receptor molecule, which is SCF-TIR1-AFB और ABF भी उसको बोलते हैं, SCF-TIR1-AFB, it gets separated, the dimer gets separated. This ARF IAA dimer will get separated. So what is this SCF-TIR1-ABF? Now let's understand this. This SCF is SKP Culin F complex, F box complex, sorry, F box complex. This is SKP, C for Culin and F for F box protein. So this is SCF protein, SCF protein complex rather. Next is TIR1. TIR1 stands for transport inhibitor response 1. TIR1. This is transport inhibitor response 1. And third is ABF or otherwise called as AFB. Both way you can call it ABF or AFB. This means auxin signaling F box. Auxin signaling F box. Okay. So this is SCF TIR1 ABF or AFB. This the function of this protein complex is to initiate the degradation of the repressor protein, which is AuxIAA protein, via the process called as ubiquitination. This part, this is called as ubiquitination process, where the targeted protein will be degraded through the process of ubiquitination. If you want to understand the ubiquitination process, please comment down below. Now, auxin SCFTIR1 AFB protein and this repressor, that is AuxIAA, will form one complex. Okay, here we can see this part. AuxIAA is there, auxin is there, SCFTIR1 AFB is also there, and these will form one complex. This complex will initiate the process of degradation. Now ARF is free. Okay, this ARF is now free and it will be active when it will come as homodimer form. Okay, so this ARF will form homodimer and it will enter into the nucleus as transcription factor and will add or will join the promoter region of the DNA, which is responsible for the further gene expression for auxin. Okay, so this is all about today's class. Kindly encourage my effort and like, share and subscribe this channel. Thank you.